Okay, applications of cryopreservation is a method which deals for long-term preservation of plant materials or animal materials, and disease-free plants can be conserved or preserved using this method, and endangered species can also be maintained using the method of cryopreservation. Pollens can be maintained to increase the longevity. Rare germplasm and other genetic manipulations can be stored. Suitable combinations of cryoprotectants and regimes of cooling and rinsing allows successful cryopreservations of biological materials such as semen, embryos, oocytes, ovarian tissues, testicular tissues, etc. Examples of cryopreservation. First example is embryo cryopreservation, and it is used for embryo storage when in vitro fertilization has resulted in more embryos than is currently needed. Pregnancies have been reported from embryos which are stored for more than 16 years. The results have been uniformly positive, with no increase of birth defects or developmental abnormalities in case of embryo preservations. Second example is the ovarian cryopreservations. In this case, ovarian tissues cryopreservation is of interest to women who wants to preserve their reproductive functions beyond the natural limit, or whose reproductive potential is threatened by chemotherapy. For example, in hematologic malignancies or breast cancer, the procedure is to take a part of the ovary and perform slow freezing before storing it in liquid nitrogen. Oocyte cryopreservation. Human oocyte cryopreservation is a new technology in which a woman's egg and oocytes are extracted, and then these are frozen and stored. Later, when woman is ready to become pregnant, the eggs can be thawed, fertilized and transferred to the uterus as embryos. Semen cryopreservation is another example of cryopreservation. In this, semen can be used successfully and indefinitely after cryopreservation. Longest reported successful storage is 22 years, and it can be used for sperm donations where recipients want the treatments in a different time or place, or as a mean of preserving the fertility. Testicular preservation is another example of cryopreservation. Here, cryopreservation of immature testicular tissue is a developing method to avail reproduction of young males who need to have gonatoxic therapy. Health offsprings have been obtained from the transplantation of frozen testicular cell suspensions or tissue species. Significance of cryopreservations, as with the examples which I have mentioned previously, these are also indicating about the significance of preservations. Some other preservations are as follows. Significances are as follows: cryopreservations of gametes, embryos, that prevent the genetic drift. It safeguards the genetic integrity of valuable strains. It also offers generations times and allows further contributions of genetics, and it eases the transportation of genetic stocks, and it also decreases the disease transmission. Thank you.